Sometimes there'll be a researcher, let's call him Larry, who does some research and comes up with this really novel approach, and that approach ends up being a valuable product, and the world is better because of it. Sometimes that person doesn't have a lab and access to the reagents and the pedigree of training, but dabbles. Sometimes that person has lots of training and dabbles. But instead of having a private company or lab, that person has access to like the world of resources through something that's open source. And we live in a time where innovation is increasingly democratized. I studied that a little bit at MIT for a year, and it's amazing. Where you give the population access to whatever it is, software, reagents, data, and they do things with it that you probably wouldn't have achieved if you waited for the one star to come along. So I'm a big fan of open source everything, open source antibodies, and so is Dr. Jim Trimmer. And he's going to talk to us about that magic right now. Thank you.
Thanks, Dave. Thanks to Larry's for this incredible opportunity at this tremendous symposium. To Meredith for handling all the logistics. I'm going to be talking today about work on antibodies. And we've heard a little bit about proteomics, and I'm going to be talking about this in the context of spatial proteomics. And specifically as it relates to mammalian brain and our work that has focused on developing reagents to enable the pursuit of spatial proteomic studies in mammalian brain to try to gain insights into really the molecular details of what constitutes a healthy brain, how that's disrupted in disease, and potentially identifying targets that could serve for the development of therapeutics to address the great unmet need in addressing brain disorders. I'm going to be talking about work that was performed, primarily, I'll get the hang of this in a second, in my laboratory at the University of California, Davis, and we've been incredibly fortunate and privileged to be funded by the National Institute of Neurological Disorders and Stroke for virtually my entire career, and more recently by the Brain Initiative. And I'll be talking quite a bit about the Brain Initiative today, and these are components of the National Institutes of Health. And I just wanted to take a second and remind ourselves of the mission of the NIH. It's a really honorable mission to seek fundamental knowledge about the nature and behavior of living systems, and then applying that knowledge to enhance health, lengthen life, and reduce illness and disability. And I also want to talk about the mission of academic institutions. So I work at a public academic institution, the University of California, where our mission is teaching, research, and public service. And today I'll really be focusing on two aspects of that mission, research, as well as the public service mission. And one aspect of that is making its research results available for public use and benefit. And both of these types of institutions, the University of Colorado is another stellar member of the academic research enterprise. Both of these types of institutions are under tremendous attack, as many other pillars of what we've come to recognize as liberal Western democracies. But I really encourage you to really take to heart contacting your elected officials, talking to your fellow citizens, and doing whatever you can. And again, most of us are at a loss of what we can do on a daily basis, but to really embrace the value of this and really think about how we can continue to have opportunities to benefit from institutions like these. I'm going to be talking today about the brain, so I just full disclosure here, I'm a very basic scientist. I'm what would be referred to in the vernacular as a wet lab bench scientist. Okay, so I'm going to be talking about things that are a little bit out of my sphere of expertise, like open source software, and especially in a room full of computational experts. And also, I'm not a clinician, so and I'm not putting this slide here to then contrast it to Nelson's and say, well, your disease has this impact on society, and the brain disorders, which is what I study, has another larger, smaller, equivalent impact. But we all know that brain disorders have a substantial societal impact and really still represent a tremendous unmet need for both diagnoses and treatment. The numbers are really staggering as far as the cost of direct health care. It's increasing with an aging population, and I was reflecting during Nelson's talk when I was a lab tech before I went to grad school. I worked in a cancer lab. And my boss at the time said, if you have a healthy heart, you're going to die of cancer. And I think now, right, we can add, if you have a healthy heart and you don't die of cancer, you're probably going to end up with a neurodegenerative disorder or some other form of a brain disorder, neurological type. And again, it's highly prevalent worldwide. And then combined with that, as far as what we lump together as brain disorders, and again, these are not separate, right? So cardiovascular disease can affect brain health, vice versa. Mental health illness also affects a large portion of our population. Fortunately, there's been tremendous progress in what we know about the brain, and it's really advancing at a tremendous pace, in large part because of the Brain Initiative. So this was initiated by President Obama in 2013, and it's really brought together researchers from all different disciplines to really begin to address, primarily through the development of innovative neurotechnologies, the structure of the brain, its activity, and how we can use this information to intervene, diagnose and then intervene in brain disorders. This was just from a few weeks ago, one example of a major advance from the Brain Initiative, which is mapping the activity and the ultrastructure of a portion of the mammalian brain. So this is cerebral cortex, incredibly complicated, but these researchers were able to do live cell imaging in an awake animal using activity reporters and record the activity from a volume of the mouse cerebral cortex, and then pass that sample along to researchers at the Allen Institute, where they did serial electron microscopy, cutting 30,000 sections, ultra-thin sections from this, and then reconstituted the entire volume of that portion of the brain, and were able to then reconstruct the 200,000 cells and their 523 million synaptic connections. Okay, so the circuitry in this region of the brain. Now, this is an incredible accomplishment. It was thought to be impossible even a decade ago, but it's still a very small volume. So this is a millimeter cubed, which is about the size of a grain of sand. So we still have a long way to go there, and this is really looking at function and structure, anatomical structure. And really, for a lot of the development of novel neurotherapeutics, we really need molecular information. And kind of a little bit of a segue to this afternoon's session, we really need to be thinking about the molecular heterogeneity of the cells in the brain, and also their subcellular compartments. And this is where spatial proteomics comes in, because it's really all about location, 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 not only of the cells and their circuits, but the molecules within these cells that are mediating these functions. Again, as a consequence of the Brain Initiative about a year and a half ago, a very comprehensive map of the entire mouse brain was accomplished. And again, another set of papers in Nature. And through single-cell RNA sequencing, which we heard a little bit about earlier today, 500 different cell types were identified in the brain. Sorry, 5,000. And almost all of these are neurons. And the brain contains maybe 4 or 5 times as many cells as the liver, but the liver is composed of 5 cell types. So the cellular complexity of the mammalian brain is immense. As part of this study, the researchers took canonical members of these different types of cell types and used them in situ hybridization, labeling of mammalian brain sections. This is really a survey, so they cut 10 micron sections every 200 microns apart, labeled them for these RNAs, and came up with this spatial map. So now this is spatial transcriptomics. What are the messenger RNAs that characterize these cell types, and how are those cells distributed within the brain and the different brain regions and the different circuits? Again, really important for identifying the molecular heterogeneity of the cells and how they map onto the structure of the brain. But what we really need is information at the protein level. And in the brain, this is really important because of the complex morphology of brain neurons. So these neurons extend these very long processes. And this really underlies this function of the neurons because function is compartmentalized within these subcellular compartments. So we have the dendritic arbor here that extends out from the cell body or soma. And the soma is the part of the cell that looks like a lot of the other cells in our body. It's got the nucleus, the ribosomes, the endoplasmic reticulum, the Golgi apparatus. And then in addition, neurons have these processes. The dendrites are the receiving end of the neuron. The other neurons form synapses on here and project chemical information onto these that's then converted into electrical information at these chemical synapses. The cells also extend very long axons that are going to then form synapses onto their target neurons. And it results in the fact that while messenger RNA is most likely to be present in the cell body, where transcription translation is occurring, the protein can be anywhere. And what this means is when you look at a brain section and you measure RNA levels, if you think about an RNA that's localized here, expressed in CA1 layer of the hippocampus, which is where I've put this RNA marker here, if the protein's in the dendrites, it's likely to be found very near the RNA. Simple case. However, if the protein is out here in the presynaptic terminals, the protein could be in a completely different brain region. And in a mouse brain, it could be a centimeter away. In a human brain, it could be tens of centimeters away, or even a meter away if it's a projection neuron that's projecting down to your spinal cord. And you really can't tell where that protein's going to end up by looking at its sequence. So having the RNA information is critical, but really we need protein information. And this is exacerbated further by the fact that this simple schematic of this neuron doesn't do justice to the morphology of real neurons. Here's the soma. This is a dendritic arbor. This is shown in 2 dimensions. It's obviously 3-dimensional. We can see the fractal structure here, similar to, so the dendritic arbor comes from a tree. And what's really important here is that these different branches of dendrites are performing different functions because this neuron might be receiving input from 30,000 other neurons. And these neurons are making synapses onto different branches. So the neurons that are synapsing on here are mapped onto these dendrites. These dendrites contain ion channels that can amplify or dampen the electrical signal that's generated at the synapse. So we're really having a tremendous amount of filtering of information from different regions of the brain, from different sources, different distances from the soma. And the soma is the site where the action potential is going to be generated at the axon initial segment. So again, this spatial information is really crucial to understanding the role of different molecules and arguably their potential value as drug targets. So a lot of work's been done to begin to map the subcellular localization of proteins within mammalian brain neurons. And one of the things that's come out of this is sometimes the localization can be incredibly restricted. So there's processes in place in neurons that even though virtually all the protein in the cell is being made in the soma, it can end up in incredibly restricted subcompartments of the cell. So here's just a few examples. These are synapses, so this green protein, virtually all of it ends up highly concentrated at these very small puncta, maybe half a micron across, that represent synapses. Here's 3 different proteins that have been labeled in white here that are trafficked to these presynaptic terminals. This is a node of Ranvier of a myelinated axon, which is going to be conducting the action potential down to the presynaptic terminals. These proteins are really precisely localized. They're segregated from one another and segregated from the entire rest of the neuron where they can perform their functions there. And this is an axon initial segment. Proximal portion, distal portion, this can represent maybe 0.1% of the surface area of the neuron, yet virtually the entire population of these proteins that's produced in these cells is trafficked there and retained at these sites. And we really need this information to really begin to think about what the impact of a drug would be on that protein, because if the same exact protein or a very similar protein present near a synapse on a dendrite would be performing a very, very different function than that same protein at a presynaptic terminal, and therefore a drug targeting that would have very different effects. In addition to the fact that a drug may be acting on a protein whose messenger RNA is here, but if it's on the dendrites, the drug will be affecting that brain region. If it's in the terminal, it'll be affecting that brain region. About 20 years ago, my colleague Ken Rhodes, who's been in the pharmaceutical industry his entire career, and I wrote an article on how we could use antibody-based validation of drug targets to really assist in evaluating the potential of different proteins expressed in neurons as their potential for drug targets. Again, the antibodies are critical for identifying the localization here as specific probes for proteins. And this is a really important aspect of drug development at the initial critical stage that's really the fundamental basis of this, which is target validation. So can we predict which molecules, if we developed effective modulators of those, might go on to serve as effective pharmaceuticals at this early preclinical stage? And obviously, if this information is not reliable, then the foundation is weak. And it doesn't matter how good of a modulator you developed, it's not going to have the intended purpose. So we focused a lot of this work on antibodies because they really are the most commonly used reagent for doing protein level research. And we heard from Nelson about Larry's amazing work, Larry and Nebojsa and others at SomaScan, developing these wonderful aptamers. But antibodies remain the most widely used reagent for doing proteomics research, and they're used in just a variety of different applications. For spatial proteomics, the primary application is immunohistochemistry. Okay, so this is a technique where samples are taken from a tissue and they're fixed to preserve the structure and also to prevent their putrefaction. And then imaging, labeling is done of specific molecules, and then imaging is done so that we can really identify the distribution of organizations within the tissue itself. And this is going to provide us with information on their structure, and it really should be done in 3 dimensions. Historically, this is all done on fixed brain sections, so in mammalian brain, fixed brain sections about the thickness of a piece of paper, and labeling with antibodies and then imaging with microscopy. One of the issues that's been raised, and we raised this initially back in 2006, is that oftentimes if the antibodies that are used are unreliable reporters of protein localization because of a lack of specificity, especially in immunohistochemistry. And this has subsequently been identified in preclinical cancer research as a problem, more broadly in biomedical research, And unfortunately, this is not just 10 years ago, 20 years ago. It's still a problem that there's many laboratories that are struggling to perform the necessary research because of a prevalence of poorly performing antibodies that are plaguing the pursuit of research. So when I started my lab, I'm interested in voltage-gated ion channels, these proteins that carry electrical signals within neurons. And the molecular identity of the voltage-gated ion channel gene family was just starting to be identified through molecular cloning studies. It turns out it's a very large family. It's the third largest family of signaling proteins after GPCRs and protein kinases. And these also represent the three major families of what's referred to as the druggable genome. But we had little information on the protein characteristics of the members of this family because of a lack of probes. So we set out to develop probes using the information from DNA sequencing, expressing recombinant proteins, purifying them and using them to immunize mice to develop monoclonal antibodies. So monoclonal antibodies is a pretty mature technology now. It's about 50 years old. And the technology comprises isolating B cells, the antibody-producing cells, from immunized animals. And the problem with those is you'd like to isolate individual cells and culture them and recover the antibodies from them, but they don't survive in culture. Kohler and Milstein figured out that you could fuse these cells to a cancer cell, and cancer cells thrive in cell culture. You can grow them pretty much forever. And you could do this to generate immortal hybridoma cell lines secreting a single antibody, because in B cells, the genetic process occurs of allelic exclusion such that each individual B cells only makes one type of antibody. They can make up to 5,000 copies per second, but it's only one type of antibody. These are phenomenal factories for making antibodies. You can just culture them in standard mammalian tissue culture at any scale. And importantly, you can archive these by preserving them in liquid nitrogen. So we can take cells that we developed 30, 35 years ago in liquid, that are stored in liquid nitrogen, pull them out, put them back in culture, and they'll be making the same antibody they were back then. And as such, they represent a really important renewable resource so that we can reproduce experiments that were done long ago using the same antibodies and extend the results from that. And this really is a major aspect to enhance research reproducibility. So we set out to develop a pipeline specifically to develop antibodies for spatial proteomics and mammalian brain. One of the things that we did was we took a large number of candidates and included steps that really focused on antibodies that would work on these fixed brain sections. And one of the issues with fixation is it's a harsh chemical treatment, and it changes the chemistry and structure of the target proteins, so that antibodies that might have been developed for these other applications where they're widely used might not work, and oftentimes don't work on these types of samples after these chemical treatments. So we included steps in our screening process that really focused us in on identifying antibodies that would work there. You get what you screen for is the mantra in screening. And then set up technologies where we could do this at a fairly high throughput. One of the reasons that the problem with antibodies was initially identified was when knockout animals became available and people did immunohistochemistry on their brains, the signal that was present in wild-type animals didn't change in the knockout. So it wasn't target specific. So we include, as much as we can, knockout screening. So these are the red antibody labeling here in wild-type animals, completely eliminated in the knockout, and then the green antibody is a counter stain just to show that the samples are of adequate quality. And then the other thing we include is just analyses of whether these antibodies work in human brain. So we develop all these using a platform that employs rodent brain for practical purposes. And you can see here two different antibodies we've developed, one that can be transferred into studies in human brain and one that's only working in rodent brain. Over the course of 30-some years of generating these monoclonal antibodies, we've generated a large library, most of which came after we started working in conjunction with the NIH back in 2005 to really try to apply our screening pipeline to develop reagents to benefit the broader neuroscience community. And our targets came from NIH as being high priority targets for neuroscience research. And we've made all of these, and the top antibodies from every one of these projects available through a low-cost distribution system. And one of the really key components of this was not only the NIH subsidizing the development of these antibodies, but the University of California contributing by not charging any licensing fees or royalties. So that's helped us keep the costs down. A large number of samples have been distributed, resulting in the savings of tens of millions of research dollars. And we think the impact is even more because they're highly validated. It saves a lot of waste of staff time, getting incorrect results, wasting samples, and then the ripple effect of not having this unreliable information in the literature. We track research publications, provide all these on our website, as well as detailed protocols, et cetera, to really inform our end users. One of the things we've also done is make the hybridomas available. So the hybridomas secrete the antibodies. Typically, you're buying the ready-to-use antibodies from commercial suppliers. So this is the primary means that people obtain antibodies for use in research labs. The typical cost of a vial of antibodies that contains 100 micrograms is about $400. It can approach $1,000 for a vial. We've partnered with the Developmental Studies Hybridoma Bank at the University of Iowa to not only make ready-to-use antibodies available at a lower price, but also make the hybridomas available. And once you have the hybridomas available, you could outsource these. We use a company in my lab that provides us with 50 milligrams of antibody for $800, so that brings the price down substantially. But if you want to grow your own, which again is very simple, can be accomplished by most research labs, we can bring it down to 10 or 20 cents relative to the 400. And just as a comparison, this is a therapeutic monoclonal antibody. So these are some of the most expensive treatments on the planet. And commercial research reagent antibodies are way more expensive than therapeutic antibodies. So this is a very valuable way to democratize accessibility to antibodies for labs that don't have large amounts of research funding. But another thing that is able to contribute to this is, or where this can make a contribution, is in projects that require large amounts of antibodies. So historically, as I said, immunohistochemistry has been used for spatial proteomic studies, cutting thin sections of brain sections, labeling them and imaging them. But a decade or so ago, a technique was developed where intact tissue could be made transparent, so through tissue clearing methods. So here's a brain before and after being cleared. So you can see now it's transparent. And these now, with advances in microscopy, specifically light sheet microscopy, advanced image processing, other computational approaches, as well as just the capacity to store huge amounts of data, have allowed for imaging of intact brains at high resolution. So these are blood vessels in an intact mouse brain. And this has led to a very ambitious program through the Brain Initiative to try to label and image 100 intact and cleared human brains. So this is now going to represent a large number of different individuals, different ages, healthy brain individuals, as well as patients with dementias, epilepsy, other neurological disorders. And the issue here, right, is first one of scale. So it takes approximately 2,500 times the amount of reagent to label a human brain as it does to label a mouse brain. And this represents about 40 milligrams of each antibody. And if we extrapolate that out to 100 brains and 50 or 100 antibodies, it comes out to a cost of antibodies alone of $800 million to $1.6 billion. So this is obviously untenable for a program whose total funding is high at $90 million, but doesn't approach this. But the 4,000-fold tenfold cost savings that we get from the types of open access antibodies we're supplying will help to make this program more practical. Another thing we're doing is converting our antibodies into recombinant form. And there's many advantages as far as standardizing the use of antibodies that come from this that have been highlighted recently. It makes the archiving much more simple, makes them much more easier to disseminate, and also turns them into molecularly defined reagents. So you know exactly what you have in the vial because you can sequence the plasmid that's being used to produce the antibody each time you're going to use it. And also, very importantly, you can further engineer these through recombinant DNA technology to really customize the antibodies to your own needs. So individual researchers can do this in their own labs, assuming they have access to all of the components that comprise the system for making the antibodies across the board. We've undertaken a high throughput sequencing. We developed a platform for high throughput sequencing, hybridomas. The antibody sequence now about 8,000 of our hybridoma samples, and made all of this information public on this publicly accessible website, the NeuroMab-Seq website. And it contains detailed sequencing information as well as information on each antibody. And then we've also used this information to develop expression plasmids so we can take the variable domains that really define the specificity and efficacy of each antibody, pop them into a expression plasmid, and recreate these recombinant antibodies, which then we validate in side-by-side comparisons with the conventional antibodies. And then made all of the plasmids available through the nonprofit plasmid repository Adgene. So we've deposited about 700 plasmids there already for different antibodies. Adgene started distributing these. And we've also partnered with them to make ready-to-use antibodies because, frankly, many people don't want to produce them on their own. They just need something that they can use in an experiment. But what this represents is, for the very first time, researchers can obtain the ready-to-use antibody, the plasmid that encodes it, and the sequence from one source. And having this available really started me thinking about analogies to open source computer software. So having this all available in an open source format, it really, the analogies are machines that execute programs written in software, machines that execute programs written in the genetic code, and making this information available to the community is really expanding the opportunity for people to really understand the reagent that they're working with. And this leads to great enhancement, more eyes on it, looking for vulnerabilities, greater customization for end users, and importantly, being cost effective. And also just making this available internationally, democratizing the availability of antibodies. So it's not only well-funded researchers in privileged countries that will have access to this, but due to the low cost and ease of producing these in any laboratory that can perform these basic techniques, really allowing for access to these antibodies for various uses to again, advance research across the entire biomedical endeavor. So we just published a call that really to encourage researchers to first take advantage of what we've developed and also to contribute to them. So if you have antibodies that you've developed, to really consider converting them into recombinant form and making that information and those reagents available, again, to enable more reproducible, transparent, and cost-effective research in the community at large, and also to encourage funding agencies to support the further development of these resources. And again, with the goal to use these in studies like spatial proteomics research to identify targets for drug discovery efforts, and also to further basic knowledge as related, in our case, to brain disorders, but other unmet medical needs for basic biomedical research. So I'll happy to end there. there, and I'd like to really acknowledge this is just the tip of the iceberg of people that more recently have contributed to our efforts at the Neuromab facility and in my ion channel research lab, core facilities at Davis that helped us develop our sequencing platform, our great colleagues at AdGene and the Developmental Studies Hybridoma Bank, and then really highlight these tremendous public servants at NIH. They're really dedicated and have really helped us over the years in coming up with concepts, innovative concepts, while making sure that we're being reliable stewards of the taxpayers' money and really trying to translate the initial investment that NIH has made for the greater good to make these reagents to have their highest impact. More recently, again, through the BRAIN Initiative, and again, I'd like to acknowledge my university for their support. And thank you, and I'm happy to answer any questions you might have.
That's great. Thank you. We're going with where the microphones are. Oh, yeah. If you'd like to submit a question through Slido and you're in this room, if you haven't figured it out already, you can point your camera at this QR code, click on the link, and then if you want to, you can anonymously enter a question.
Yes.
I guess this is not an anonymous question then. So I'm curious. I think this is really important work that as a community we should be doing. My main question is really, how do you incentivize companies to actually start doing that, right? It's already hard enough individually within an academic institute.
Yeah, it's a great question, and I have to say, 20 years ago, I really thought that, companies our antibody companies were doing a horrible job, and there were quite a few that were. They have really, a number of them have really come around, but they have a very difficult challenge because they maintain really large catalogs of antibodies, each of which at the initial stage is an investment on their part that they have to recoup through sales. And, I know they make an effort to maintain inventories even of antibodies that aren't high sellers if they're critical for individual research labs. But again, they're in a business, and at some point, too often those antibodies are withdrawn from the market, and because they're licensed, they're really unavailable. So it's really a tough thing. And I have to say, right, I'm an academic researcher who's been funded by the NIH, whose work has been subsidized. So I'm really in a privileged position to be able to do this, and I'm happy to do it. But where's the line where a company is putting themselves at risk by contributing to something like this? And I've had discussions with them, and they said, you're making your sequences public. Once that genie's out of the bottle, it's gone forever. And there's going to be companies that are going to be pirating these and selling them for profit. And then the question is, well, do I want to stop that and penalize the whole rest of the research community? Or are we just going to deal with that as a cost of democratizing these and kind of live with it? And I just came down on one side, but again, I'm coming from a place of being able to do that, which many folks aren't.
This world needs more people who espouse that philosophy from the bench through clinic and epidemiology, and I really admire that. Thanks.
Thanks, Dave.
I've been told that we have time for one more question, because two more questions.
Two Larrys, two questions.
Because we have lunch ready, so yes. Please state your name just so people know who you are.
Oh, yeah. My name's Larry Hunter. So I love that. And I actually don't have a question about your talk. I just love it. And I hope someday that all the SOMAmers that we made at SomaLogic are equally available, because they're different in ways. But that's not my question. My question, because I knew most of what you said from you, and the question was in your second or third slide. I almost fell out of my chair. There really is a Genentech facility that makes that amount? Will you say something about that?
Yeah, I mean, you know, unfortunately, it's only 20 miles away from Davis, and unfortunately, I've never visited there. But I know people that work there, and, you know, it's like a biochemistry lab at 1,000, you know, it's like Honey, I Shrunk the Kids, but the other direction. You know, every column's like this big around and, you know, 100 feet long, ion exchange, huge bioreactors, producing all these therapeutic monoclonal antibodies and other biologicals, recombinant insulin, et cetera, for use throughout the world and therapeutically. It's just remarkable, and the level of detail that goes in from starting with a vial of cryopreserved cells and expanding that, and all the quality assurance and quality controls that goes into making sure that, you know, kind of like over in Golden with the Coors factory, right? You know, how do you make beer that's like that at that scale? And you know, you just have to do it.